Hi there, my name is Andrew Brown, and welcome to Glycogenesis and Glycogenolysis. This video will run the full gamut on glucose storage in the cell. We'll start with the structure and function of the macromolecule glycogen, and then we'll cover the metabolic and catabolic processes whereby it is built up and broken down. After watching this video, you should be able to answer questions like this one, which tests your knowledge of the individual types of bonds broken and formed by the branching enzyme during glycogenesis. We should begin this review of energy storage with glycogen, the polymeric form in which glucose is stored. Firstly, these granules, which are found in the cytoplasm of animal cells, are structurally complex. A protein at the center, called glycogenin, connects hundreds of molecules of glucose in chain form. Secondly, the production and breakdown of these granules occurs in liver and muscle cells, where liberated glucose is either used to maintain blood glucose levels in the liver, or fund metabolic activity in muscle cells. Now that the structure and purpose of glycogen has been exposed, we're going to take a look at the buildup and breakdown of glycogen granules. As we introduced already, the glycogen molecule has at its center the protein glycogenin. When glucose monomers are added to a growing glycogen granule, phosphate sources like uridine diphosphate and triphosphate, along with inorganic pyrophosphate, will be used to convert the form of glucose used in metabolic processes to the form necessary for building glycogen. Recall from what you know about cellular respiration that glucose 6-phosphate is the first intermediate in glycolysis. A mutase will convert glucose 6-phosphate into glucose 1-phosphate. Remember, mutases are often used to rearrange functional groups. The glucose 1-phosphate then continues along, its activation being coupled with uridine triphosphate dephosphorylation, producing a uridine diphosphate glucose dimeric intermediate. And after the action of glycogen synthase and a branching enzyme, which decouples the molecule of uridine diphosphate, we end up with glycogen. It's helpful to see glycogenesis distilled to such a simplified pathway, but in order to fully understand what's going on here, we should take a closer look at those last two enzymes. The first, glycogen synthase, is a rate-limiting enzyme that adds molecules of glucose to the growing glycogen chain using alpha-1,4 glycosidic linkages. When you hear rate-limiting step, an alarm should be going off in your head. They're prime MCAT fodder, and rightly so. What better place to impose a control point than the exact step in which glucose molecules are being converted into their storage form? In fact, the glycogen synthase checkpoint is deeply integrated with other elements of metabolism and even digestion. Insulin and adenosine triphosphate will serve to stimulate glycogen synthase activity, whereas epinephrine and either glucagon or adenosine monophosphate, depending upon cell type, will inhibit it. Now, what about that branching enzyme? Glycosyl alpha-1,4 alpha-1,6 transferase, don't worry, its name is a bit outside the scope of the MCAT, takes a linear glycogen chain and introduces branch points. The way it does this is by breaking some of the alpha-1,4 linkages and reconnecting the freed oligoglucose segments elsewhere with alpha-1,6 linkages. The more branches that are created in a molecule of glucagon, the greater the efficiency with which energy can be stored. Now think about that for just a moment. Remember, we are dealing with biological systems here. So as the latter enzyme is making branch points, glycogen synthase is operating as well. As a result of the creation of more branches, you actually get an effect reminiscent of Le Chatelier's principle, where more terminal glucose molecules are being made available for glycogen synthase, allowing both enzymes to operate closer to their maximum rate. That just about wraps up glycogen metabolism, so the next logical step is to take a look at glycogen catabolism, or glycogenolysis. Starting with glycogen, stored either in liver or muscle cells, a series of enzymes convert back to glucose 1-phosphate, which is then just a rearrangement and a dephosphorylation event away from glucose. Similar to glycogen synthase in glycogenesis, glycogen phosphorylase serves as the rate-limiting step in glycogenolysis. It will phosphorylate peripheral alpha-1,4 glycosidic linkages, creating glucose 1-phosphate monomers as the linkages are severed, but will stop at the branch points. More on that in a moment. Quickly note here that glycogenolysis is regulated in the opposite fashion as glycogenesis, with signs of ample free energy inhibiting the enzyme and signs of depleted free energy stimulating it. Glycogen phosphorylase was able to liberate glucose from the linear sections of glycogen, but in order to deconstruct the branch points, we will need a second enzyme, or rather two more. The debranching enzymes, glucosyl alpha-1,4, alpha-1,6 transferase, and glucosidase, then work in conjunction to break alpha-1,4 glycosidic linkages at the branch points, form new terminal alpha-1,4 linkages, and then hydrolyze the leftover alpha-1,6 linkages. This last step, performed by glucosidase, is perhaps the most noteworthy, as it releases individual glucose molecules directly, the only example of such in all of glycogenolysis. As glucose 1-phosphate molecules are released sequentially from glycogen chains, the same mutase used in glycogenesis converts them back to glucose 6-phosphate. At this point, we are again reminded that this process is occurring in a biological system. The glucose 6-phosphate produced from glycogenolysis can directly enter glycolysis, the metabolic process whereby glucose is converted into two molecules of pyruvate. That is only one option, however, because glucose 6-phosphate can also be converted into regular old glucose by the enzyme glucose 6-phosphatase. Speaking of glycolysis, recall that glucose 6-phosphate was the result of an irreversible reaction there. Well, it's actually the location of the glucose 6-phosphatase enzyme that allows that statement to hold up. Because the enzyme is embedded in the endoplasmic reticulum membrane with its active site pointing into the lumen, glucose 6-phosphatase will not be available to react with any of the intermediates from glycolysis. That concludes the material this video is going to review. Now let's employ what you've learned on some sample questions. This first question is testing your knowledge of the types of bonds made or broken by the branching enzyme in glycogenesis. The answer should be C. Questions like this one, where a complex biological reaction is simplified to answer choices with increase or decrease of various products, are MCAT favorites. To get the answer quickly, try to be as simple as possible about the action of the enzyme. Using that strategy, the branching enzyme decreases the number of alpha-1,4 linkages while increases the number of alpha-1,6 linkages. Because the question is asking about the effect of decreasing branching enzyme activity, just look for the opposite of those phenomena. This next question is asking about the function of glucose 6-phosphatase. The answer should be D. The first step in getting this question is locating glucose 6-phosphatase in the catabolism pathway. Because the enzyme converts glucose 6-phosphate to glucose, the immediate effect of defective glucose 6-phosphatase would be reduced blood glucose levels, eliminating answer choices A and B. Anytime an enzyme is knocked out in a multi-step reaction, a backup of reactant is created, in this case, glycogen. If you got those questions correct, awesome job. If not, please feel free to watch this video again as many times as you need. Before we finish, however, let's go back and look at the entire glycogenesis and glycogenolysis processes one more time. In glycogenesis, we followed a molecule of glucose as it was phosphorylated, rearranged, ligated to uridine diphosphate, and then rearranged again to make glycogen. We then noted how the buildup of glycogen stores is regulated either directly by levels of free energy or indirectly by hormones and neurotransmitters. Glycogenolysis, then, retraced this pathway, enzymatically reducing glycogen back into glucose 1-phosphate and eventually glucose 6-phosphate or glucose. We looked at this process's regulatory mechanisms as well, which are essentially just the opposite of the mechanisms utilized in glycogenesis. The last element, and this is more related to how the process fits into ongoing metabolic processes in the body, showed how the outputs of glycogenolysis can feed directly into glycolysis or glucose regeneration. Thanks very much for watching, and good luck in your continued MCAT study.